Hello, and thank you for joining us today. Our webinar is Advantages of TOC over the HPLC for Cleaning Validation. Our presenter today is Tyler Trent, Elemental Product Marketing Manager. This webinar is being recorded and will be available within a few days. Okay, now I'm going to turn it over to our presenter, Tyler Trent.
Thank you for the introduction, Shelley. As Shelley stated, today we're going to be going over the advantages of TOC compared to maybe some other techniques out there for cleaning validation, such as HPLC. So the first thing that we kind of want to talk about is, what does TOC consist of? What is TOC for some that may not know? Well, TOC consists of living organisms, decaying matter, even synthetic organic products, coal, plastics, and other items as we go through. But how does this balance out to what TOC means? TOC means total organic carbon. Well, if we remember our chemistry, there's a lot of different types of carbon out there. We have inorganic carbon, we have non-purgeable organic carbon. So how does this fit into the building blocks of what a TOC analyzer can truly measure? Well, here's kind of like a balancing scale of what TOC consists of versus what inorganic carbon, IC, consists of. So when we kind of look at the values here, or we look at these sums, if we get TOC plus IC, we get total carbon. Well, what does that really mean for us, or how do we really do this? And a lot of the laboratories that we're working within today, especially in the pharmaceutical realm, really care about total organic carbon. The inorganic carbon could be purged off, and so that gives us our certain value. So once again, in recap, total organic carbon means it's a measurement for monitoring all organic carbon pollutants in water. So it's really a catch-all for carbon. So we're not looking at specific compounds or specific analytes. We're looking at carbon as a whole. How much carbon is present into our samples? So when I talk about flow charts for carbon, we start off with the total carbon. Total carbon up top here, this is what our TC value is. This is total carbon. Total carbon is made up of inorganic, IC, and also TOC. Well, a lot of times is when we are looking at TOC, we're really only looking at this realm right here. It's called non-purgeable organic carbon, or NPOC. So what does that really mean? Well, we basically take our sample, we add acid to it, we acidify it down to roughly a pH of 2, maybe 3, and we sparge that sample. What does that do for us? It's going to drive off our inorganic carbon. It's going to release it, because we don't want to measure that right now. We want to measure our non-purgeable organic carbon, or what we like to say is TOC equals NPOC. So that's going to give us our non-purgeable organic carbon, which is what we're looking for our TOC measurement. Just to reiterate, TOC can be broken down into 3 simple steps. Well, first we got to remove the inorganic carbon. We have to oxidize the sample, and then we have to measure the sample after the carbon has been converted over to CO2. So we already talked a little bit about the removal of IC. Now let's talk a little bit about the oxidation techniques. There are 2 primary oxidation techniques out there. First is a combustion. It uses a catalyst in a high temperature oven. In mixture with oxygen or air, and it performs a oxidation reaction there. But then when we start looking at the other way we can do this, this is done by the UV persulfate oxidation. A sample is placed inside the UV chamber with sodium persulfate solution, and then the sample interacts with the UV light and causes oxidation. Well, those are our 2 techniques, but how do we measure our CO2 after it's been converted from carbon? Well, on the systems here at Teledyne Tekmar, we use a system called a NDIR, non-dispersive infrared. It's basically we have our sample gas, our CO2 will pass through, the infrared, the IR, will be absorbed by the CO2, and that will give us a certain type of reading. From that certain type of reading, we can take that and correlate it against the calibration curve, do our check responses, and get a true value for carbon, for our TOC, or as we like to call NPOC. There's another form out there where it's a membrane conductivity or conductivity detector, and this uses a liquid phase as it passes through a membrane. We won't go into much detail on that, but note that Teledyne Tekmar uses the detector called the NDIR. So what are some of the characteristics that we're looking for in this type of analysis? So within the pharmaceutical realm, we're going to be utilizing a UV persulfate. UV persulfate is the system of choice when it comes to ultra low levels, clean water samples that will be facing or coming across in the pharmaceutical market or pharmaceutical field. So what are some of the characteristics that we must be looking for? Well, we need to have low system lengths because carbon's everywhere in this world. Our water, there's carbon. Our DI water, there's carbon. Even if we're looking at water for injection, WFI, there is a certain amount of carbon that can be in that we have to test for. So we need a low system length. We're not going to need anything that can contribute to a high elevated response. So that's why combustion is typically not the system of choice or the oxidation method of choice because it does have a high contribution factor for system lengths. We need to find something that is excellent precision at low ppb levels. Characteristics of UV persulfate, it doesn't handle particles. That's why this is so predominantly used in, say, a pharmaceutical market or a clean water market, because it's going to allow us to get the utmost oxidation reaction occurring, and we don't have to worry about the particulates in the sample. And so, like I said, a UV persulfate with an NDIR, ideal uses are within the pharmaceutical market or also within the drinking water. But this presentation, as you can see from the title, we're going to be focusing on the pharmaceutical aspect. So when we start looking at the cleaning validation, there's methods out there, USP 643, where we must do system suitability with 1,4-benzoquinone and sucrose. But there's another aspect that pharmaceutical manufacturers must do. It's also validating that their systems or their reactors or vats are clean. Well, there's a couple techniques out there that they can do this by. The first technique is high pressure liquid chromatography, or HPLC. There's a great advantage to using HPLC in cleaning validation. From an analytical perspective, it includes a lot of increased sensitivity, a lot of specificity, and robustness. If we're looking for a certain type of compound and we know that compound is going to give us that peak and we are able to develop an analytical method for that, that is great. We can find that and we can do our analysis on that. But if there is an unknown and you don't have a mass spec or you're using a UV vis and there's an unknown peak, well, it's going to take you some time to figure out what that unknown might be. Well, when we start looking at TOC, TOC is a catch-all. It's going to get everything for us. So when we start looking at HPLC and the cleaning validations, manufacturers of drugs and other biologics, there's active ingredients, there's washing ingredients or detergents for cleaning aspects. We got to make sure none of this stuff is being carried over. Well, how do we do that? Obviously, we have to test for that. HPLC is often used because it's there. They have one. Cleaning validations a lot of times will tie up an HPLC. But if you had another system that can get your cleaning reagents or agents, it can get your active ingredients, your byproducts, and other material, what if you can do that in all one type of system? Is that the only common source of potential contamination? No. And as I'll show you in another in a case study down below, you're going to have other outside factors that might contribute to this other than what you're using in the manufacturing and cleaning process. So you want to make sure that you can get everything done by one type of system, which is TOC. So, the big question is, would it be more effective to have an instrument that can test all sources of contamination? Whether it comes from a human error or your cleaning agents or from your active ingredients or even your water. You want to be able to say, yes, I want to have a system that can test for all sorts of contaminations. Well, that's kind of what TOC does. Is it compound specific? No. Can it tell me if there's benzoquinone, or can it tell me if it's benzoquinone versus another material in there? No, it cannot. But what it's going to do is it's going to tell me my carbon level. And if we go back to what carbon does or what carbon is, it's, living organisms, decaying matter, synthetics, organic products, or other material that have carbon in, because carbon is a basic building block of everything that we know. So if we look at this chart here on the screen, TOC, it's going to have a broad analysis with low-level detections. Any disadvantages? Yes, it's non-specific. But in this realm or in this world, we're actually looking for a go, no-go value. If we're above a certain value, then we need to go back and revalidate our cleaning process, send the samples back through. Oh, it's clean, we're good to go now. Or we can do further investigation where we bring in other tools such as HPLC, TLC, or other materials or other type of testing equipment. So, HPLC is very specific. You're going to be testing for the compounds that you know of. You're going to be calibrating for the compounds that you know of. It's highly specific. There are some disadvantages. You're going to have long run times. It's an expensive tool. It requires a lot of deep analytical knowledge. You got to understand what type of buffers, mobile phases, what type of column you might need, or even how to dial in the ramp parameters of how the solvent or mobile phase goes through the system. Well, with TOC, it's very simple, very easy and straightforward. So when we start looking at capabilities, HPLC can do drug residues and excipients, and then if you look at some cleaning reagents, it might struggle on some of the cleaning reagents. Same thing with thin layer chromatography, but TOC, being that it can capture all carbon, it's going to get you across the board. Yes, it's across the board. So when we're in these laboratories, we want to make sure that we have a system that meets our analytical needs in detection limits and low-level detection, but also very robust. And it's going to give us excellent capabilities across all types of sources of contamination that could be done in our cleaning validation process. So TOC has actually grown in acceptance for cleaning validation. And then if there's any further investigation, that's reserved for an HPLC. TOC methods, they develop and validate and use effectively for pharmaceutical cleaning validations. We work directly with certain vendors or certain manufacturers to ensure their IQ, OQ, PQ is set up properly, and then their system is validated, and then they run their waters for us. And then they run their waters on our system. So they run there's two steps of a cleaning validation. One, you must run your rinse water, and two, you must run your swabs. And it's a very simple process, and TOC can handle both of those sample types. As I said, over time, TOC has gained a lot of credit in the pharmaceutical industry. Here's a paper, for example, TOC has low-level detection, rapid analysis time, and low-cost comparison to other methods. When we start looking at a TOC system can be purchased for, let's just say, less than $40,000. And then when you start looking at a HPLC mass spec or HPLC UV vis, your value or the value of that system is going to be, more than $50,000. It could be depending on what type of detector you're going to be put on there. So how does TOC really work for us? How does TOC really make a difference in the cleaning validation? Well, like I said, it has a low system cost when compared to HPLC. Very quick analysis, up to 10 times faster than other techniques. Minimum sample prep, because you basically take your sample, you take your water that you get from your cleaning rinse, put it into a vial, place it into the system, and little to no method development is needed. And it's very low cost of operation, as the reagents used by the system are inexpensive. So you might have a better ROI on this, because it's going to be able to ensure your production is continuing and you're not down for certain types of contamination. You know, as I spoke about this before, it's very non-specific. TOC is non-specific, so it detects any carbon-based substance. If if there's low TOC, that's possible. If there's high TOC, the possibility of contamination is there. If it's low, possible contamination is remote. The same instrument can be used for your pure water. The newest update to USP 643, when it's talking about your storage and bulk water that you're using in your manufacturing facility for pharmaceuticals. And it's also used for water for injection when you're starting looking at injectable drugs. That water has to be below a certain TOC content. So you're getting a lot of bang for your buck with this type of system. So as we move on, we're going to move on to maybe a real world example here. So this data was actually volunteered by one of our customers. We're looking at some type of equipment, blend mix and different things like that. Well, there's the TOC results. 304 ppb, 21.9 ppm, and 96 ppb. Well, they have actionable limits. Their limits are set to 1.1, 2.0, 2.0 ppm. Well, the first one is well below our limit. Does anything need to be done? No, it doesn't. The second sample, wow, that is way above our limit. So there must be other actions that are taking place. Well, they can go through, start cleaning it again, or they can continue to go through the process and try to figure out where that contamination is coming from. So they actually went through and looked at an HPLC. They used this as a specific technique, and they were able to obtain an active peak. Where is this active peak coming from? So they continued to do more method development and understand what this peak could have been. So they took the sample again, reran it on TOC. It yielded the same results of 21.2 ppm, similar to the first, and it exceeded their established working limits. After 2 weeks of investigation, it was proven that it wasn't something in their process. It was actually contamination from hand sanitizer that the person was using while collecting the sample. So that allowed 2 weeks of investigation, and that was identified by the by the LCMS with several modifications. So this was a sample contamination and not a system contamination from their vats or other things. So it was clear that the customer saw contamination that the HPLC may not have ever found, or they would have to do further investigation to understand what that active peak was. So the TOC, allowed them, with the power of the cleaning validation, to detect contamination outside the active pharmaceutical ingredients. So you're not only just looking at what is in the active ingredients or your cleaning residues or your other material. This is actually going to capture everything, which is a good tool to use. So, we're going to talk a little bit about disadvantages, as we also talked about some advantages. Some of the disadvantages to HPLC, it's a very well-established technique. That's one great advantage to it. But when we start looking at a comparison to TOC, yes, HPLC is highly specific. TOC is non-specific because it will capture any type of carbon. HPLCs are generally used because it's available there in the lab. They're doing a lot of research and development. Other types of tests are performed on HPLC, so it's there. Also, cleaning validation or CIP samples typically tie up to HPLC. So a lot of customers might need a dedicated system or a backup system, or have other samples could be delayed. When we start looking at runtimes, HPLC as standard runtime could be anywhere between 20 to 30 minutes, depending on methods. But in TOC, you're getting a single rep done in 3 minutes. HPLC does require a lot of analytical knowledge to set up the method parameters, but it's also a very good tool. The same method is used in TOC regardless of the product. You're going to utilize one pharmaceutical method in the TOC system, and it's going to provide you the results. So, we were talking to some auditors or different things like that as we go out and speak. A lot of times is HPLC sometimes is more likely to get questioned because there's more variables, more unknowns that you can be doing with HPLC. TOC is very simple. It gives you one robust number. And if that number is higher than your actionable limits, that action is required. You either need to go back through and clean, or you have to do further investigation to understand what your contamination is. So the advantages, like I said, TOC systems cost 2 to 3 times less. Better to originally purchase TOC for CIP samples, and faster runtimes means faster turn times. So we're getting samples done quicker. Also, the biggest thing that we state is at the end of the day, TOC will provide you with a go or no-go answer depending on your established limits. So when we look at the TOC value of the swab in the rinse water, if it's below our actionable limits, we can proceed with work. But if it's above our action limit, what do we need to do? Well, certain manufacturers have certain SOPs and processes put in place. Sometimes they'll go through and repeat the wash and then retest, or they'll go into further investigation to understand why that sample is contaminated and get to the root cause. So it's a simple TOC, as I said, provides a simple go, no-go answer. So here at Teledyne Tekmar, as we talk about UV persulfate systems, we offer a system called the Fusion. The Fusion is the 5th generation UV persulfate from Teledyne Tekmar, which was previously a Dorman TOC analyzer. This actually combines the analyzer and autosampler all in one. So it's all in one package. It doesn't have an autosampler that sits off to the side like other vendors, or as an additional add-on. It's all included into the system itself. We also like to make the chemistry viewable. We also like to make sure everything is up front and can be seen. So on the front of the system, we have what we call the chemistry area. This is where our IC sparger is, where we remove our IC. We have our water and copper tin scrubber where it removes contaminants and also dries out our CO2. So moisture is not getting into the detector. We utilize a 7-port syringe. And then we have the UV reactor where the UV light will interact with the sample that contains a persulfate reagent and then oxidizes the carbon to CO2. We also have inside the system, where we want to make it easy to work on, is we also have our permutation dryer, NDIR, and our valves are very accessible by removing the side panel. We also utilize a mass flow controller. This mass flow controller does 2 things. A, it allows us to ensure our system is pressurized and has constant readback, so we know the system is not leaking and it's doing exactly what it needs to do. But it also allows us to adjust our flows and modes to speed up the system instead of being fixed by restrictors and regulators. Then we also have, as I said, we also have the auto sampler on top. This does contain a 75-position 40 ml Voa vial auto sampler rack, and it has 4 stock positions on the inside. These stock positions will allow you to do certain types of features with this system, which we'll talk about in a second. So instead of making your calibration standards by hand, you can actually make the system do it. The system will prepare by auto-calibrating from a diluting from the stock standards in those center 4 positions. It will make the calibration for you. They can also do auto dilutions of samples and standards, and you can calibrate for many different ranges. So as I talked about before, with the Fusion, we want to obtain accurate low-level TOC that maximizes carbon response while minimizing background. So that's what we're trying to do here with the Fusion. And that's what we're delivered. That's what has been so predominantly known about the Fusion moving forward in its lifetime. So characteristics of a UV oxidation system, UV persulfate oxidized system. Yes, we can handle large sample volumes. I believe the pharmaceutical method is up to 9 mils per sample, or 9 mils per rep. That gives us the utmost sensitivity and utmost detection at ppb levels. Sample is discarded after analysis, low system blanks, great precision, low maintenance. So when we start looking at this, yes, I talked about this system can be used for drinking water. So if you're looking at drinking water methods, these are what it's standardized to. But, within the pharmaceutical cleaning validations, we kind of go after the FDA, GMP, even the, USP 643, the European pharmacopoeia, the Japanese pharmacopoeia. And we also look at for water for injection, cleaning validations, and other aspects across the pharmaceutical industry with the TOC analyzer, with the Fusion UV persulfate. As you can see here, the analysis time is between 5 to 7 minutes. We can vary different types of modes. If we want to do total carbon, TOC, TC minus IC, look for IC, and different things like that. And it's just a very list of the Fusion specifications there. But the biggest things that we want to talk about with this system is some of the features that I've mentioned before. One, the auto calibration, the open view of the chemistry components, so you can look across the lab and understand that the system is doing what it's doing by looking through and seeing it. Mass flow controller, we'll talk a little bit about static pressure control in a second, and teledilution. If you're in an environmental lab, which we wouldn't be using this feature in the pharmaceutical lab, it would go back and redilute your sample and bring it within the calibration range that you have selected. The system does come with 21 CFR software. It's a standard software that comes with it. It's not an additional add-on or anything like that. At install, you can select standard instrument without 21 CFR or 21 CFR tools. We give you tools for compliance, and then serviceability and easy to software use interfaces. So as I kind of spoke about some of these features, calibration features, auto calibration from a single stock standard, you can make, and the system can do it for you, or yourself can make your user standards and run them into individual vials. You can do multi-point, linear, quadratic with auto blinking, auto checks, basically the same thing. You can do it from a single stock where the system will do a dilution from a known value, or you can make it your own by using user calibration standards. You can set pass-fail criteria. You can set decisions. If it fails, what do you want it to do? Do you want it to stop, recalibrate, continue? We give you a lot of features, a lot of tools inside the software to use. As I spoke about this before, the MFC, this allows for mode-to-mode changes the flow rates. What does that mean? That means faster. It's going to run the sample quicker. It allows for higher flows to be used to clean between samples, so that helps us reduce our carryover. And it also will turn off if the system is in standby. So you're not utilizing gas as it's flowing through the system when it's not being used. It's actually turned off and stopped at that mass flow controller until you click ready on the system itself. We use something called static pressure concentration, or SPC, for our NVIR. So this gives us better performance under pressure. So you might ask, what does that mean? Well, we take the NDIR, which is typically a flow-through system, and we pressurize it. So we close off one end. That allows the CO2, the carbon that's been converted to CO2, to build inside that detector. Once that detector or the system has reached equilibrium, typically around 50 psi, we're taking a snapshot of the CO2 and it's being analyzed directly. So it's one instantaneous reading of a very sensitive sample. So this right here kind of shows you a calibration curve. We were able to do from 10 ppb up to 1 ppm from one single standard using the auto dilution on the system. The results are displayed graphically. So you can see here on the screen, you can see you have a baseline plus blank, raw detector value, and then your adjusted detector counts. So everything is listed here for you. So when we start looking at performances and things like that, yes, we can go all the way up to 1 to 200 dilution on the syringe. But you know, we were working down at a 5 ppb level. Please note, this is very ultra clean, very ultra trace, and you have to follow specific recommendations to try to even achieve these levels. And a lot of pharmaceutical labs are typically working at maybe 50 ppb or higher. But this is just showing off the flexibility and capabilities of the system. One thing that I also mentioned with this presentation, is that, you know, the Fusion has validation support, has an IQ/OQ/PQ. So this is an optional item that can be added to the quote or different things like that, and it comes with a full validation of the system done by a trained engineer on site over a certain amount of time. And then here you can go in and set up certain protocols, such as user managers, user permissions, electronic signatures, and a lot of different things. And also within this IQ/OQ/PQ, we're testing the calibration features, the reagent blank analysis. We're doing specificity challenges. We're looking at calibration and method validation stuff. We're looking at cross-contamination challenges, the famous system suitability with sucrose and 1,4-benzoquinone, and your reagent water, and other types of items that need to be done for the pharmaceutical environment. So in conclusion, there is another option out there for cleaning validation, and it is TOC. TOC does provide a lot of different advantages that HPLC may not be able to provide you instantaneously. With that said, you know, I would like to close out this presentation. I want to also say thank you for your time and attendance today. And if there are any questions, please let us know. And also, please be sure to follow us on our social media channels, Facebook, LinkedIn, Twitter, YouTube, and we also have a podcast called TOC, where we actually go into topics like this or talk to industry people, where they give us their advice and techniques and tips and tricks. So that's also there. And also, we started doing different types of blogs on our websites. So please be sure to follow the social medias, and you'll be able to stay up to date with everything that we're doing here at Teledyne Tekmar. Thank you for your time.